Hi everyone, so welcome to Chapter 5. In Chapter 5 we are going to be talking about beyond Mendelian genetics. So this is taking it a step further. We looked at Mendelian genetics in Chapter 4 with monohybrids and dihybrids, and now we're going to talk about the next step in beyond Mendelian genetics. So in Mendel's traits, he just looked at two different traits, so dominant and recessive. And now there are going to be exceptions to that. Most genes in our body do not exhibit the simple monohybrid, dihybrid traits, so we're going to look into more realistic examples of that. So the first example is incomplete dominance. So this is a heterozygous phenotype is a blend of those two homozygous phenotypes. So if you take a red flower and a white flower and you cross them, you're going to get all pink. So you're going to get a blend of that. So it wouldn't be showing just red if it took over. Now it's going to blend the two together. So an example of this would be familial hypercholesterolemia, or abbreviated as FH. So the heterozygote here has half the normal numbers of receptors in the liver for LDL cholesterol. So it's showing that it has half good and half bad. And then in a homozygous, the mutant allele totally lacks the receptor, so the cholesterol level is very high. So if you have a high cholesterol level, you have more chance of getting heart disease or having heart problems. So you want to keep an eye on that. But this example shows that if you have both, you would just have a high amount, and then if you have a blend, you would have a blend of half the normal and a blend of half regular receptors. So this is an example here. So in this example, in FH, individuals can have a normal, half, or 0 receptors for LDL cholesterol. If I mate together a normal male with a heterozygous female, what are the probabilities of the genotypes and the phenotypes of the offspring outcomes? So first you have to look at what you're crossing. You have a normal male with a heterozygous female, so I'll show you what the outcome would be. So in this example, now with incomplete dominance, you're going to be using all capital letters, and you're going to be using a different letter for the other trait that could be in there, not just lowercase and capital. So normal would be FF, and then heterozygous female would be F and C in this example. So then you're going to cross them, so F and C, F and F, you see what you get. All right, so you have half normal, and then you would have half with half of the receptors for LDL cholesterol. The second example is codominance. Okay, so this is the heterozygous phenotype results from the expression of both alleles separately. So back to the flower example, so if you have a red flower and a white flower, and you cross them, this time you won't get pink. This time it's codominance, so you're going to show both. Like if you would have a co-teacher in a classroom, there would be both teachers teaching, and in this example you would have both red and both white showing like spots. An example of this would be sickle cell disease. So if you have an H and an A, it would be normal shaped, and if an H and an S, it would be sticky. So either normal or sticky cell for red blood cells. And heterozygotes make both. So you would have some normal and some sickle cell types in their blood. So another example, this is for codominance. So two individuals that are heterozygous for red blood cell shape mate together. What are the genotypes and phenotypes for the example? So in this example, it shows using superscripts for the shapes of the red blood cells. So you would have a heterozygous person with an H and an S, okay, and you would have another individual person with an A and an S. I don't know if I said H, but A and an S and A and S. So they are heterozygous for the sickle cell anemia. You would cross those two. Okay, you would get an H and an A, H and an A. So you just cross them the same way. You just bring down the H and the superscript, the H and the A and the S superscript, and bring it down just like that. So you would get 1/4 with an 2 capital A's. You would get 1/2 with an A and an S, and 1/4 with an S and an S. So you would only have 1/4 normal. You would have 1/2 showing both, and you would also have 1/4 severely sick with sickle cell anemia. There are also multiple allele examples, so the individual carries 2 alleles for each gene, so multiple. A single gene can also have more than 2 alleles because it can vary. So different allele combinations can produce different phenotypes. For example, the PKU gene has hundreds of alleles resulting in 4 phenotypes. The CF gene has over 1,000 alleles. And blood typing alleles can be either IA, IB, or I. So A blood, type B blood, or O blood. So as I just said, A, B, and O gene encodes as a cell surface protein. So the A allele will produce the A antigen, B will produce the B antigen, and O will produce no antigens at all. A and B are codominant, and both are completely dominant to the I or the O phenotype. So if you're looking at this picture here, you can see that the A type blood, okay, in this, when you do blood typing, you'll see these I's here. That's how they show the different phenotypes, and then the little I means that it's O blood. Okay, so if you see an A, then it means A, or B, it means B, but if you see the little I, that means that it's O. So in this case, the A's together, they make just A, and in this case, the A is taking over the little I, so you would get A blood there because A is dominant to O. So you will only be making A antigens when you have A blood, B antigens when you have B blood, and then when you have an A and a B, it's codominant, so you would have an A and a B antigens. And then when you have type O blood, you would have no antigens because it does not make any. The only way you can have O blood is if you have 2 little I's. The only way you can have AB blood if you have an A or a B. There are 2 different ways for other blood types. So example of this, a man with type AB marries a woman who is heterozygous for type B. What are the possibilities for phenotypes and genotypes? Okay, so you're going to cross that. Okay. So you automatically know that there's only one phenotype or genotype for AB. It would have to be IAIB. But then you have to look at the B blood because you could have all capital B's or B and little I. So it said heterozygous here, so it's automatically going to be B little I because it has to be different. So then you plug that in, you cross them, you're going to cross the same way we did the last one, and you are going to get 1/4 AB blood, 1/2 type B blood because you have big B, and then a big B here is going to take over the little I, and then 1/4 A blood. All right, so blood transfusions, if a person were to receive a transfusion of blood, the type that they do not have, their antibodies would attack the transfused cells as invaders. So you can only get the same blood type that you have, unless it's a certain situation. There would be clumping, a reaction that would occur, and your antibodies would go in your immune system and attack that blood that does not need to be there. So to be avoided, you can transfuse with the same blood, or you can use the O type. That's blood is the universal donor. So you can donate blood, O blood, to anybody. Anybody can receive O blood. Or you can use type B, AB blood, that anybody can receive any blood type. So O blood you can donate to anybody because it has no antigens on it, so everyone can receive that. Or to use type AB blood, that person can receive all the different blood types because it can receive A, it can receive B, and it can receive AB and O. So they are the universal recipient, so if you're that person, you're pretty lucky. But if you have type O blood, you would want to donate a lot because everyone can receive type O blood because it has no antigens on it. So this is just for you to practice the type of transfusions that a person could get. So type A blood, what type of blood do you think that they could receive? Type O blood, what type of blood can they receive? And type AB blood, what can they receive? So we just talked about that type AB blood is the universal recipient, so they can receive all blood types. Type O blood, you have to think about what antigens are on O, which are none. So they would only be able to receive O blood because if antigens came into their body, they would attack it. And type A blood, they could receive just type A blood because if they had AB blood come in, they would attack the B. But they could also receive O. Alright, so these would be the examples, example answers. So type A could receive type A and type O, universal donor for type O. Type O could only receive type O, and then AB, AB, O. Alright, so penetrance and expressivity. Penetrance, all or nothing expression of a single gene. So you either have it or you don't. And then expressivity is the severity or extent of the gene. Genotype is incompletely penetrant if some individuals do not express the phenotype, and then phenotype is the variability. Expressive is symptoms vary in intensity among different people. So phenotypes can express differently, especially with different environments in some people, and then genotypes can be incompletely penetrant if some individuals do not express a certain phenotype. Alright, pleiotropy. It's a phenomenon where one gene controls several functions or has multiple effects. So for example, I always say this wrong, but porphyria virgate. Alright, so that affected several members a long time ago, and it's a symptom or disease where a single enzyme is missing, and then because of that enzyme being missing, it results in a lot of symptoms. So for this example, reddish teeth, photosensitivity, and then that also inspired many vampire and werewolf legends because of this disorder. So this is a little tree of how this happens. So if there's a missing enzyme, the furflin accumulates, and then this can result. So the missing enzyme, your urine can result dark red. If it's missing in the digestive system, it can result in constipation, abdominal pain, and so on. Alright, so Marfan syndrome, last example. It's pleiotropic. So one change in the gene or missing enzyme results in tall and slender build, disproportionate long arms, legs, fingers, and toes, breastbone protruding, high palate and crowded teeth, heart murmurs, and so on. So one specific enzyme or gene changed can affect a lot of different things in the body. All right, I hope you filled in your notes. If you have any questions with this, please ask me, and we'll be doing a lot of practice.